Good morning, listeners. Once again, I want to welcome you to my YouTube channel. I remember my humble self, Dr. Mkujo channel. So, a very quick one. Today, we're going to be looking at a very important parasite. Don't mind me. In this channel, we'll be talking about parasitology, and of course, what else can we talk about apart from parasites? So, we talked about entamoeba isolatica as a parasite, and we have also talked about Giardella and Blair. So, they want to look at a very important parasite that is called Trypanosoma sp. So, follow me. So, Trypanosoma sp. Why am I saying sp? I will say explain. We have different species of this particular parasite. Mm? Now, we have Trypanosoma brucei complex. And why are we saying brucei complex? Because inside this brucei, we have three other subspecies. As a matter of fact, we have Trypanosoma brucei, Trypanosoma brucei rhodesiensis, Trypanosoma brucei gabiensis. So these are the three subspecies under one single complex called Trypanosoma brucei complex. Apart from Trypanosoma brucei complex, we still have Trypanosoma cruzi. We still have Trypanosoma equipadium that is non-transmitted venerally in horses, through sexual intercourse in horses. This one does not require a vector. So this is one of the species of Trypanosoma that does not require an insect vector, Trypanosoma equipadium. But our emphasis today in today's class is going to be on the Trypanosoma brucei complex. Now, look at the classification. You have the phylum as Euglenozoa, we have the subphylum as Mastigophora, we have the class Zoomastigophora, we have the order Trachomatida. Then there's a suborder that is called Trypanosomatina. Trypanosomatina. That is the suborder. Then we have the family Trypanosomatidae. It ends with AE. They have the genus Trypanosoma. And all these species I've not mentioned now, Trypanosoma cruzi, Trypanosoma brucei complex, Trypanosoma equipadium, and so many other species that we can't exhaust here. Okay, now characteristics of this parasite. Number one is that they are hemoflagellate. Now, in protozoa as a whole, we have three main types of flagellates. We have the one they call the intestinal flagellates. It means it dwells, is a flagellate. And what do we even mean by a parasite being a flagellate? It means that it uses flagella as its locomotive structure, as simple as that. So we have some intestinal flagellates. That means they dwell in the intestine. The example is Giardella and Blair. We have the tissue dwelling flagellates. It means they dwell in the tissue. Example is Trichomonas vaginalis. They will now have the hemoflagellate, which we are talking about now. So the first characteristics of Trypanosoma is that they are hemoflagellate. That means they dwell in the blood. They stay in the blood. They live in the blood. And don't forget that this Trypanosomatina, which is a suborder, is a kind of suborder they share with another sister parasite, which is called Leishmania. But maybe in subsequent classes or lessons, we may look into Leishmania. Have a Topica, Donovani. We have several species of that Leishmania. So they share the same order and suborder with this Leishmania. 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 Okay, now I've talked about the classification. Then it's also important you know that we also have two different groups or classes of Trypanosoma. We have the one they call the salivarian group. We have the one they call the sacrovarian group. The salivarian, sacrovarian group. Now, when we talk about the salivarian group, this group of Trypanosoma, what they do is that they inhabit or live in the fore and the midgut of the insect vector. Don't forget, the insect vector is sessile. The biological name is Glossina. And we have many species of this Glossina which we'll look into. So The salivary group, they dwell within the midgut and the foregut, and transmission is through inoculation. When the insect bite, you know, they transmit the parasite through injecting it into the host body. That is inoculation. Then the Saccharine group. This group is a group that stay within the hindgut, and in this case, transmission is through fecal contamination when the insect bite. So, having known this now, is important to now go into the life cycle of this parasite. Before going to the life cycle of this parasite, one thing I want you to understand. Okay, I've not even finished the catalysis. Don't mind me. Uh, Another characteristic of this parasite is that they have a flagellum that arises from a basal body that is called kinetosome. Kinetosome. It arises from a basal body, a basal body. That is where the flagellum arises from. It's called kinetosome. Then, method of division, it multiplies through simple binary fission. And in this binary fission, maybe I'm going to treat that in details in another class. It means that a parasite divides into two daughter cells, and that is the method they use in multiplying. It begins with the division of the nucleus. After the division of the nucleus, which is called karyokinesis, then the cytoplasm now divides, which is called cytokinesis. So these are the two different events that happen in binary fission. So another important characteristic is that they have different morphological body shapes. We have the one they call the epimastygote. We have metacyclic trypanosoma. We have opisthmastygote. We have amastygote. We have choanomastygote, and so on and so forth. These are the different morphologies. So this parasite, has the ability to change into different forms during the course of its life cycle. We're going to look at some of them. I'll see some of them in the course of the life cycle, discussing the life cycle. So let's go back to the life cycle of this parasite. Now, coming to the life cycle, the infective stage is the metacyclic trypomacygote. Please don't forget, the infective stage is a metacyclic trypomacygote, and it's usually found within the salivary gland of the insect. So when the insect now ingests this infective stage into the body of man, then it first of all changes into slender trypomacygote. And this slender trypomacygote will first of all now multiply at the point of inoculation, and subsequently can multiply in the blood, can multiply in the lymphatic system, within the tissue fluid, and at the later stage of the infection, it can multiply in the CNS. And this parasite is also even taken to the heart, where it also multiplies. Now, after this multiplication, then the next thing that happens is that the insect vector, which is the fly, will now come and pick up the parasite, the trypomacygote in the blood. Once it is picked up, they multiply in the midgut of the insect, and within 2 to 3 weeks, it's being taken to the salivary gland of the insect. Once it gets to the salivary gland of the insect, that is where it now changes into another morphological body form, or transforms into another form, which is now called epimastygote. From this epimastygote, it changes into another body form, which is now called metacyclic trypomastygote, and this is the infective stage. So it remains in this salivary gland until when next the insect has a blood bill, or bites another person, then the cycle continues. So this is the life cycle. So you can look at it the way it is in this diagram. Metacyclic trypomacygote injected into man by fly, then multiplies in the blood, in the lymph, and in the CNS. Then trypomacygote is taken up by the fly. Then this trypomacygote changes into epimastygote, and then epimastygote into another into the infective stage, which is now called the metacyclic trypomacygote, and of course the cycle continues. Now, the next thing to talk about is the pathology. What does this parasite cause? What are the effects of this parasite to the host? Now, this pathology, I'm going to divide it into two different stages. We have the early stage of the infection, and we have the late stage of the infection. At the early stage of the infection, that is when the person begins to experience a headache. The person may even feel pain. Okay, the first thing is the earliest sign is inflammation, swelling at the point of inoculation, and it's called the chancre. The chancre. The person, sorry, the person experiences inflammation at that point where he or she was bitten by the insect vector, that is chancre. So that is the first thing that happens, the earliest sign. Then before you now talk about the, the headache, the person may begin to experience headache. Then of course swollen lymph node. Now we have different places in the body where the lymph nodes are located. We have at the back of the neck, we have within the lower part of this jaw, we have lymph nodes here, and it can be inflamed. Then at the armpit, we have lymph node. Then within the groins, we have lymph node. These are the different locations in the body where one can experience one can see the lymph node. So this parasite also goes to inflame the lymph node. Then this swollen lymph node is what we call the lymphadenopathy. Now, if you have swelling or inflammation at this part of the body, it's not lymphadenopathy. The lymph nodes are located within this region, then within the armpit and within the groin. That is where you see the lymph node. Now, at the late stage of this infection, don't forget I mentioned early stage and late stage. At the late stage of this infection, that is when this parasite gets into the CNS. When I say CNS, I mean the central nervous system. And once it gets to the central nervous system, it causes what is called meningoencephalitis. What do we mean by meningoencephalitis? It means the inflammation of the meninges. Meninges, that membrane, the membrane that covers the brain and the spinal cord, or have the dura mater, or the pia mater, or have the arachnoid mater. So these are the three membranes. So it goes to inflame it. And the inflammation of this membrane is called meningoencephalitis. The person also experience trembling, sleeping sickness. This sleeping sickness that people know with this disease, even when the person is walking on the road, the person is sleeping, if the person is eating, the person is sleeping. So that is sleeping sickness. And the trembling, weight loss, the person will also begin to experience weight loss or apathy. The person is no longer interested in anything. This happens at the late stage of the infection. Treatment. How can you treat this disease? Very simple. Take blood sample, then use microscopy or use a microscope. So the method is through microscopy. Why do we use blood sample? Because the parasite dwells in the blood. And what stage of this parasite can you see in the blood? These trypanosomes. Can you see the? Remember I said during the course of the life cycle that once the metacyclic trypanosote is injected into the blood, it changes into a slender trypanosote, which now multiplies in the blood, lymph and whatever. So if you check the lymph and the blood, what you're going to see is the trypanosote. That's the stage you're going to see. It's an exam question, so students should be careful of this. So that is what happens. So coming to the treatment, like I said, we use two drugs for the early stage. Pentamidine for the infection that is caused by Gambians, use pentamidine. Then for the one that is caused by rhodesians, use suramine. Then at the late stage of the infection, if the infection has gotten to the late stage, these two drugs cannot go through the barrier of the CNS to kill the parasite. So what we now do is you take or use a more potent drug, which is now called melarsoprol. Melarsoprol is a more potent drug that has the ability, that sustains the ability to permeate through the membrane of the CNS. So that is for treatment. Then epidemiology, spread of the disease. How does this disease spread? Of course, the simple way through which it spreads is through its insect vector, the glossina. Now, we have two endemic areas. You can see this glossina. We have the riverine area, we have the woodland area. desert sort of, where you have less of water and as the case may be. So within this region, you see, within the riverline area, you see Gambians, you see more of Gambians, and you see Tachinoids is one of the vectors that can spread Gambians within this region, and for sipes. Then within the woodland region, then that is where you see more of rhodesians, and you can see mosquitans and the palidives. So we have other species like a swany tawny and what have you. So we have different species of glossina that can, or fly that can help to spread this particular disease. Prevention and control, very simple. You can use the natural enemies of the vector to reduce the population of the vector. Then you can also use insecticide to kill the parasite. Then of course, bushes around the houses should also be cleared to destroy the breeding site of the parasite. And several other things you can do, like also detecting the infection early enough. It can also help to treat this disease. So I think we've done justice to this topic. So I'm gonna end it here. Please share this video to as many as you know that offer this course, parasitology, medical students, and other people that may need it. It will help them a lot. Thank you so much. I remember my humble self, Dr. John Uchenna. See you in the next class.